Hello everyone! Today we're going to learn about the auditory system after the visual system. The auditory system is another exemplary sensory and perception system. In this episode, we're going to first take an overview of audition and then take a look at the sensory organ, the ear, or more specifically, the cochlea. In the next part of this episode, we're going to take a look at the brainstem nuclei, the middle geniculate nucleus, which is part of the thalamus, then the primary auditory cortex, higher cortical areas, and information processing. Let's get right down to audition. Audition is our fancy way of saying hearing. What we're hearing is sound. Sound is the stimulus, and it is in fact a type of mechanical vibration. To put it even more specifically, this kind of vibration is a kind of longitudinal wave. It is a wave that goes back and forth with compressions and rarefactions, as shown in this figure. There are several properties of this wave. First, the frequency. This is the pitch of the sound, whether the sound is high or low. Usually, our hearing range is from 20 to 20,000 Hz. Another property is the amplitude, which is the loudness of the sound, whether it's loud or soft. And the third property is more complex. This is the wave pattern, which is the tone. A pure sound, such as one emitted by a tuning fork, would be a pure sinusoidal wave. However, the instruments that we play or the voices that we speak are usually complex waves. They have little peaks and big peaks, and that constitutes the tone. Some other information related to sound are about the source of the sound. We would also like to know the direction of the sound source as well as the distance of the sound source from us. This is the map for the auditory system. On the left, we have a flowchart of how auditory information flows in the auditory system. On the right, we have a map of all auditory-related structures in the nervous system. First of all, the ear. Let's take a look at the structure of the ear. Generally, we can divide the ear into three parts: outer ear, middle ear, and inner ear. The outer ear is here. It is for receiving sound and funneling it inside. The middle ear is here. It is for conducting sound and amplifying it so that it is easier to hear. And the third part is the inner ear, which is used for transduction. It transduces the sound signal into a nervous signal. The outer ear. The outer ear consists of the auricle or pinna, which is the part that you see on the outside. It is directional, meaning that it helps us receive sound from one specific direction, namely this direction. This will come in handy later when we're trying to decide the direction of the sound source. The second portion of the outer ear is the auditory canal. It is about 2.5 centimeters or 1 inch in length. It goes inside our skull to reach the middle ear. Here, the middle ear. The first part of the middle ear is the tympanic membrane, which is also colloquially called the eardrum. It is a very thin membrane that vibrates with the air or whatever that is around it. The eardrum is connected to little bones called ossicles. There are 3 ossicles in the middle ear. And eustachian tube is a tube that connects the middle ear cavity with the nasal cavity. Let's take a closer look at the 3 ossicles. Ossicle comes from the word meaning little bone in Latin. The first ossicle is connected to the tympanic membrane. It is called the malleus because it is shaped like a hammer. The second one is connected to the malleus, and it is called the incus, the anvil that lies with the hammer. And the third bone is called the stapes, which looks like a stirrup. The stapes has a flat end that is connected to a window on the cochlea, part of the inner ear. This window is called the oval window. Let's assemble them together, and we get the picture of the middle ear. What would happen if there were no ossicles? If we connected the tympanic membrane directly to the oval window, then actually about 99.9% of the sound energy would be lost or reflected if the air is connected to the cochlea directly. That is because the cochlea is filled with fluid, and it is very hard for the air to vibrate the fluid efficiently. Therefore, The three ossicles serve a vital function in amplifying the sound. The middle ear amplifies the sound in two ways. Firstly, if there is vibration here at the tympanic membrane, then the malleus is vibrated and it serves as a lever to move the incus. The incus is longer than the malleus. The malleus and incus are like a lever and they together move the stapes. The stapes moves like a piston in and out of the oval window. Here, the force is amplified by the lever mechanism. Moreover, the tympanic membrane has a much larger surface area than the oval window. Since the oval window has a smaller surface area, the pressure is now even greater. Put together, these two features causes a 20 times pressure increase from the tympanic membrane to the oval window, which amplifies sound by a lot. There are two muscles that are connected to the ossicles. One is called the tensor tympani muscle, which is connected to the malleus. The other is called the stapedius, which is connected, of course, to the stapes. These four muscles play a role in a reflex called the attenuation reflex. What is this? Let's take a look at the attenuation reflex. When we hear a very loud sound, the tensor tympani and stapedius muscle contract immediately, automatically. This helps us adapt to high-intensity sounds. Now that the two muscles are contracted, the ossicles cannot vibrate to a very large extent. This decreases the amplitude of the signals, and of course, protects us from harmful loud sounds. If the sounds were too large, then our cochlea, our inner ear, would be damaged. So this is a very useful mechanism. However, please note that this attenuation reflex still has a 50 to 100 millisecond delay, so the protection from sudden loud sounds such as explosions is limited. Therefore, we still need to protect ourselves and get away from those sounds before they happen. Another usage of this reflex is adapting to high-frequency sounds. The attenuation reflex suppresses lower frequency sounds more than higher frequency sounds. When the muscles contract, the malleus and incus are more rigid, and they cannot vibrate at a low frequency very easily. However, they can still vibrate at a rather high frequency. And this is why it can help us hear speech in noisy environments. The noise in the environment is usually low frequency, while human speech is usually higher frequency. So this is how you can hear a friend when you're at a noisy party. It is hypothesized that this reflex can also suppress sound from our own speech. When I'm speaking myself, my voice is conducted through my skull into my ears. But when I hear others speak, the voice is transmitted via the air. Therefore, this reflex can help us suppress the sound of our own speech. The inner ear, the final part of the ear. In the inner ear, there is this vestibular organ, which we can see is like a labyrinth. There are many loops here. And we also have the cochlea, which is the shell-like part here. It's kind of like the shell of a snail. The shape of the cochlea is spiral. There are usually about 2.5 turns in the whole cochlea. So much for the ear. It's question time. Please select the correct statements. Note that there may be multiple answers. A. The eustachian tube connects the inner ear to the nasal cavity. B. If the tympanic membrane is broken, then it will almost be impossible to hear anything. C. Vibration is conducted from the eardrum to the cochlea by a bone called the ossicle. D. The auditory pathway directly involves brainstem nuclei, just like the visual pathway. Please take a few moments to pause and think about it. Here's the answer. The only correct answer is B. For A, the eustachian tube does not connect the inner ear to the nasal cavity, but rather the middle ear. For B, if the tympanic membrane is broken, then it will not conduct sound very efficiently, and it will be almost impossible to hear anything, unless the sound is large enough to vibrate the cochlea directly. For C, vibration is conducted from the eardrum to the cochlea by 3 bones called the ossicles. They are, in the order of conduction, the malleus, the incus, and the stapes. And for D, the auditory pathway does directly involve brainstem nuclei, but the visual system doesn't. Therefore, this statement is false. Let's take a closer look at the cochlea. This is the organ where sound transduction occurs. If we take the spiral shell of the cochlea and then unfold it, then we will get a long thin stripe, just like this. At the beginning of the cochlea stripe, there is the oval window. As we've mentioned before, it is connected to the stapes, one of the ossicles. The oval window leads to the scala vestibuli. Scala means staircase in Latin. Scala vestibuli basically means climbing a staircase away from the vestibules. Here, just below the scala vestibule, there is the scala media. The scala media lies between the scala vestibuli and the scala tympani, which we will show in a second. At the end of the scala media, there's a little hole called the helicotrema. The helicotrema connects the scala vestibuli with the scala tympani. Going from the helicotrema back to the beginning of the cochlea, we have the scala tympani. The scala tympani approaches the tympanic membrane as it goes. And then at the end of the scala tympani, we have the round window, as opposed to the oval window. The round window is the end of the cochlea's passage, while the oval window is the beginning of the cochlea's passage. Now let's take a transection of the cochlea and take a look at the structures that we've just mentioned. Here we have the scala vestibuli. Separating the scala vestibuli and the scala media, we have the Reissner's membrane. On the other side of the Reissner's membrane, here is the scala media. The scala media is bordered by the basilar membrane on the other side. And on the other side of the basilar membrane, we have the scala tympani. These are the three major cavities and the two main membranes inside the cochlea. Now, all these cavities are in fact filled with fluid. This is a zoom-in graph of the three cavities. Perilymph is the first type of fluid that exists here. It is shown in blue. Perilymph is in the scala vestibuli and in the scala tympani. It is kind of like a normal extracellular fluid, where there is high sodium ion concentration and low potassium ion concentration, just like most places in the brain. On the other hand, we have the endolymph. It is shown in green here. It only exists in the scala media. We will talk about this in the next episode as well, as it also exists in the vestibular organs. For now, let's just remember in the scala media we have the endolymph, which is a kind of special fluid. We say that it is special because it has low sodium ion concentration and instead high potassium ion concentration, much higher than the normal concentration. This special kind of fluid is produced by the stria vascularis. Stria means stripe, and stria vascularis is a stripe of tissue that borders the scala media. We can see that inside the scala media, on the basal membrane, we have a bunch of cells that are rather uniquely organized. This is the organ of Corti, and he is the protagonist of our play today. The organ of Corti is analogous to the retina. It is the organ that senses the stimuli and transduces it into a nervous signal. The organ of Corti has the modiolus on one side. This is the part in the middle of the cochlea spiral. At the bottom, as we mentioned in the last page, there's the basilar membrane. Here are some connective tissue, including rods of Corti and the reticular lamina. On top of the organ of Corti, we have the tectorial membrane. As you may recall, there is a structure in the midbrain called the tectus. Tectus means a lid. So tectorial membrane here means a lid that covers the cells that we are going to mention next. They are the inner hair cells and the outer hair cells. They are covered by the tectorial membrane. When sound comes into the cochlea, it moves the perilymph inside, and in turn makes the basilar membrane oscillate or fluctuate. The fluctuating basilar membrane then causes the hair cells to experience relative motion with respect to the tectorial membrane. We will see how this leads to the transduction of auditory signals. The hair cells are our receptors here, analogous to the photoreceptors in the eye. How do they sense the fluctuation? Well, we will take a look at the hair that they have in the next slide. At the bottom of the hair cells, there are synapses that are made with neurons in the spiral ganglion, which is also called the cochlear ganglion. The neurons in this spiral ganglion then carries information away to the auditory nerve. Auditory nerve is part of cranial nerve 8, which is the auditory vestibular nerve. So this is the pathway of output from the ear, spiral ganglion cells. Here is an electron micrograph of the organ of Corti. Let's zoom in a bit. You can see some V-shaped things here. If we zoom in even more, we can see that there are little hair that project from a cell. These are the hairs of hair cells. Hair cells sense sound via these hairs. These hairs are stuck into the tectorial membrane. When the tectorial membrane and the hair cells undergo relative motion, the hair cells are bent in one way or the other, and this causes a neural transduction. If these hair cells die out or are damaged, like this, then it is likely that some degree of hearing loss will occur. If all hair cells are destroyed, then the person will become deaf. Zooming in into one single hair cell. At the top of this image, we have a purple hair cell, and at the bottom, we have the axon of a spiral ganglion cell, which makes a synapse with the hair cell. The hair cell conducts the information to the spiral ganglion cell by releasing neurotransmitters. In other words, this is a chemical synapse. The largest hair on the hair cell is called the Cochlea. It is a stiff piece of hair that has a lot of actin filaments in it. Moreover, there are some proteins called tip links between the hairs. Tip links are rigid, rod-like proteins that connect the hair to each other. Moreover, the tip links are connected to mechanically gated ion channels. These ion channels are in the family called transmembrane protein-like family of proteins. When the hairs bend in one direction or the other, the tip links from adjacent hairs will pull or push the mechanically gated ion channels and thus open them or close them. The hair cell movement causes ion channels to open or close. These ion channels are permeable to potassium ions. When they open, there is increased influx of potassium from the endolymph. When they close, there is decreased influx of potassium from the endolymph. Here, this is the normal condition where hair cells are unbent. There is some degree of inflow of potassium because some of the channels are open. If we bend the hair cells towards the kinocilium, then the tip links pull open the mechanically gated ion channels. There is more influx in potassium ions, and the cell fires action potentials more frequently. If we bend away from the kinocilium, then some of the open ion channels are pushed close. Therefore, there are less action potentials here. How do these hair cells generate action potentials? Well, basically, they also use the voltage-gated calcium ion mechanism. Here is the molecular pathway. First, there is influx of potassium. This depolarizes the cell and causes voltage-gated calcium channels to open. This leads to calcium ion influx and causes the snare proteins to release glutamate into the synaptic cleft. Glutamate is an excitatory neurotransmitter and causes the spiral ganglion cell to fire. This is the whole process of transduction in the cochlea. As we mentioned before, there are two types of hair cells, outer hair cells and inner hair cells. There are more outer hair cells than inner hair cells. However, only 5% of the spiral ganglion neurons connect with outer hair cells. The rest 95% all connect with the inner hair cell. Why is that? That is because outer hair cells carry out an important function. They serve as cochlear amplifiers. One mechanism by which they amplify sound is through motor proteins. For example, a motor protein called prestin responds to sound and amplifies it. It kind of listens to the sound that occurs and tries to imitate the sound, emitting a sound by itself. Therefore, the sound is amplified. Another possible mechanism is by myosin and other tip link proteins. They may also contribute to the amplification role of the outer hair cells. After the sound comes into the cochlea, the outer hair cells amplifies the sound and makes the signal more receivable for the inner hair cells. This has applications in clinical situations. For example, if we want to test the hearing of a newborn baby, he or she cannot respond to us. We can use the cochlear amplifier mechanism of outer hair cells to test whether the hearing is still intact in the newborn baby. We emit a sound into the ear and then try to receive the amplified sound from the outer hair cells. This is rather weak but still receivable by our apparatus. If the outer hair cells emit a responding sound, then the amplification mechanism is likely intact. The death of hair cells is the main reason for deafness. If there is death of inner hair cells, then auditory transduction will simply not occur. If there is death of outer hair cells, then the amplification mechanism will fail, and it will be very hard for us to hear the sounds. Therefore, one approach in the case of deafness is to use a cochlear implant. These cochlear implants directly stimulate the spiral ganglion cells in order to substitute the hair cells for auditory transduction. A final point about the cochlea is pitch sensitivity. We have seen this illustration before. This is an expanded view of the cochlea. The basal membrane has several properties. It is narrower and stiffer at the base, near the oval and round windows. In contrast, it is wider and floppier at the apex, near the helical trema. The narrow and stiffer part is more sensitive to high-frequency sound, while the wider and floppier part is more sensitive to low-frequency sound. Therefore, from the base to the apex, we have a preference for decreasing frequency of sound. This is tonotopy. As we've mentioned in the previous episode, there is a retinotopy in the visual system. Similarly, there is a tonotopy in the auditory system, where the map is not based on the retina, but rather based on the tone or the frequency of the sound. This is the map. We have unlocked the first part of the map, which is the hair cell in the cochlea. In the next part, we are going to go deeper into the auditory system. Question time. Please select the correct statements. A. Inner hair cells amplify vibration so that outer hair cells can receive information better. B. When mechanically gated potassium channels open on the hairs, the flow of potassium ions is opposite to what we usually expect in most neurons. C. Hair cells at the base of cochlea generally sense high-frequency sound. The correct answer are B and C. For A, it is actually outer hair cells that amplify vibrations so that inner hair cells can receive information better. For B, indeed, mechanically gated potassium channels in the hairs allow potassium ions to flow in, which is the opposite to what we usually occur. For C, the hair cells at the base of the cochlea respond to high-frequency sound, while those at the apex generally sense low-frequency sound. In this episode, we had an overview of audition. We learned about the structure of the ear and talked about the transduction of auditory signals in the cochlea. We will get to the brainstem in the next part of this episode. Looking forward to seeing you again.